Hi, so my name is Nicola Ferrier. I'm a computer scientist in the Math and Computer Science division at Argonne. And I'm going to talk today, I have a number of projects that sort of focus around things to do with images. And I'm going to talk today about a project called Small Worlds. It's funded by the DOE BER, which is Biological and Environmental Research. And so it's an unusual project in that BER is funding computer scientists to do some imaging work. And so, sorry, I had a funeral on the weekend and got to fly the red eye back. And I've picked up a cold, so I'll try not to cough too much on you guys. All right, so one of the challenges with imaging, I looked at kind of the attendees here. There's people that do optimization, people that do various other algorithmic things. How many people here work with image data routinely? One or two, yeah, quite a few. It's hard these days not to. So I'll try not to make too many assumptions, but one of the challenges is trying to see things at smaller and smaller scales. And if you're following along, you know that there's all these new super resolution techniques and things people are doing to image at lower and lower scales. And the DOE actually, is very interested in looking at some of the biological systems, ones of interest to them. And they would like they challenged us to actually develop imaging technologies that would allow us to image live, moving cells and cellular processes. And so here you actually see, this is actually a tree root, and this is a bacteria around the roots. And there's an interesting relationship between aspen trees, Pseudomonas bacteria, fungi, and other stuff that happen around the root system. And one of the projects that has been developed at Argonne is looking at this root system. So we're using that as our test bed. And what we'd really like to be able to do, I'm going to focus today on trying to get into this scale here. This blue is where we're going to be focusing today. So there's a lot of techniques out there, and I was going to give a brief overview, but realized that would be most of my talk talking about other people's imaging technology, so I'm not going to do that. But there's confocal microscopy, STED, STEM. If you follow any of the super resolution, there's a number of techniques. And we'd like to be somewhere in that space where we can actually image these bacteriums and actually ultimately get into cellular processes in smaller scale. So our challenge was they wanted to develop a 3D imager that could look at moving cells. So we refer to this as volumetric snapshot imaging. And volumetric means you want to capture a whole 3D volume in a single shot. And snapshot means we want to get it at one time frame. And so if you know anything about the various imagers like confocal and storm, palm, STED, all these other things, they have to scan. So you actually have to fix the cells, which is the euphemism for you kill it. And plasticize it and freeze it, and so it doesn't move, so that you can do this imaging. So this isn't really conducive to live cell imaging if you have to kill it to image it. So our challenge was, we want to do things at moving cells, and things move at very different rates. So we're right now focusing on certain levels, and this was a novel space for me to work in. I come actually from computer vision and robotics, so this was kind of interesting because I was going to get to learn a lot more optics than you would think someone in computer vision might already know. So what I'm going to do today is talk a little, very, very briefly about sort of traditional methods that people use to get at three-dimensional information, and then talk about how we have been developing this imager and how we're using some of these techniques to actually do this multifocal microscopy. So very, very briefly, for all of you, here's like many years of research in 3 slides. There's a lot of ways that people have used to get three-dimensional information from images. And so you can look at things like shading. So shading of things gives you three-dimensional information. And this was circa 1980s. Monumental failure, it only works in very idealistic cases, not on anything real. You can look at texture. The way texture gives you a lot of information about surface shape. Or you could look at focus and defocus. And our eyes actually do this. So as you're not aware of it, it's very subconscious, but your eyes will change focus, and that information will tell you a lot about three-dimensional information. And this is something we will come back to in the talk. You can also use stereo. Our eyes are stereo, although people see 3D, and there's something like 8% of the population actually doesn't have stereo vision. So, but stereo, you know, if you move the camera or have multiple eyes, and you can, you know, look at an object point here. One of the caveats of stereo is you have to recognize that this object point here projects to various image points. There's all sorts of tools you can download off the web. Visual SFM is one of them. That you can just take a camera like your iPhone, take a bunch of images, crank it through this machinery, and it'll give you a point cloud of 3D data of that. And so I believe this one actually isn't. This is from Photo Tourist, which is from Washington University, their software. But so stereo works really nice, or multi-view. And again, we'll be talking today a little bit about multi-view. And then the other thing that people have used is structured light or structured illumination. So there's commercial products like the David 3D scanner, where you project onto an object, and then you take an image, and if you know lots of stuff about the pattern you've projected, you can pull three-dimensional information. And so a nice example here that's very simple, that's actually used in a lot of industrial systems to do sort of process control, is you shine a sheet of light, and if everything's nicely calibrated, and you look in here, you can see the profile that the camera would see of where the light hits the objects, and the height in the image is equivalent to the height of the object, so you get 3D information. And then if you move the objects around, you get 3D models. So structured light, again, is something that we are starting to look at. So these are techniques from computer vision, and they're all at a macro scale. But what can we use from this? Actually, to be honest, it's now, I think, closer to 60 years. Larry Roberts' PhD thesis from MIT in 1964, I think, was the first computer vision real piece of work. And I don't know how many of you know the history, but Seymour Papert from MIT, who was a roboticist, gave one of his graduate students the task of put a vision system on the robot this summer. And like 60 years later, we're still trying to get somewhere. So it's always interesting that people thought this was a really easy task, maybe because our eyes do it. So we can use focus, we can use multiple views at the smaller scales. We can use structured light. I'll allude to that very much at the end. And what's nice about working in this space is, unlike when you walk around a room with a camera or do things in industrial settings, you can actually engineer your specimen. So there's a whole line of work on developing reporters or fluorescent tags that actually help you solve the correspondence problem or figure out what you're looking at. So you can light things up with different color lights, or with, there's mechanisms where if two molecules get within so many nanometers of each other, they light up. This is called FRET. So you can develop reporters or develop sensors that help you with the imaging task. So some of the things that are harder in the computer vision space and the microscopy space are a little easier. So what I'm going to talk about is what we've been developing over the past now 3 years, and are continuing to work on, is a multifocal microscope to do this snapshot 3D imaging. And so again, a little biological motivation. If we image this, these are insulin granules in, I'll have a slide later that explains some of the biology, but this is a 2D slice. And you can see things move, and you don't really know what happens to them because things in cells are moving in three-dimensional space. So this 2D movie doesn't really give us a very good information about what's happening in 3D. Likewise, we can do some of these other techniques like the confocal scanning. And this is a Z-stack, so stepping through in depth, getting very good resolution of the insulin granules and microtubules, so the space. But we only can get one level of depth at a time, and so if things are moving, again, we won't see them, but that allows us to look at structure. So neither of these, there's this time and axial dimensional trade-off that you have to make when you're deciding to image with most conventional techniques. And so we decided we needed some of these pieces together. We needed 3D information, and we needed a snapshot so we could actually see things moving in 3 dimensions. So your conventional microscope, very crudely sketched here, has, you have a sample, you have a lens, an objective, and it projects onto some sort of camera or detector. And what usually happens is at any one time, in the sample here, you have some plane, and on the camera, that's the single plane of focus is all you get. With multifocal microscopy, what you have is, if we look at the sample here, we have a bunch of different depths. And this is a conventional microscope on this side, but then we've added this diffractive optical element that splits the beam and effectively creates 9 or 25 is what you'll see today, or you can tune that and change it. But a bunch of different images. So if you look at these, here we have something that's at 4Z, 3Z, 2Z, and they map onto the detector these different, I'll call them tiles, that are different depths in the sample all at one time. And what you'll notice, though, is that in here, on the detector then, we've given up some XY resolution because we have now 9 tiles where before you had a single wide field microscope, but we now have 9 tiles. But we have this depth encoded image. We have different depths all at one time. So depending on how you set this up, you can get different thickness of sample imaged all in one frame. So this is what it looks like. So this, if you're a wide field here, here, if you just had wide field, you would just see, this is a 170 nanometer bead. All of the tiles would look the same. But if we tune for different depths, then what you see here are, different blurriness because you're looking at different depths. The bead is only 170 nanometers, and we're stepping through there's a 3 micrometer there. So you get these out of focus images of the bead. And you might say, well, what use is that? Well, if you go back to what we talked about depth from defocus, these out of focus information, or out of focus images actually give us information about the depth. So somehow encoded in here in the size of these rings, and this is where exactly in the 3-dimensional volume is that bead. And so in a single snapshot, we can image, say, a bead here. And just for those of you, you can actually etch these diffractive optical elements. There's a great optimization task I'll get to in just a second. So there's a great optimization task in designing these, and that we have a really crude brute force algorithm that looks at how to design them and spits out what's our optical efficiency. And so we know that we have a good task here for those optimization folks of how to actually optimally design these. Yes?
So is this process related to like what the litro field camera does?
No. Actually, I have dozens of litro field cameras. So the light field cameras, that's not quite true. In a sense, the light field cameras, they take multiple, they do actually take, but more than just having different focal depths, they actually are also sampling the space from different directions. So they're more like taking a bunch of cameras on an array and taking, so they do have different focal, so there is some focal pieces in there, but we're imaging the exact same space at different focal properties, whereas the light field, you're really getting different rays. So you're getting sort of stereo built in there too. So great question. Okay, so just out of curiosity, that's what this thing actually looks like. And so if we have multiple beads here, if you have multiple beads, you can see that you can look at these, and there's just a few things I'd like to point out here. So if you look over here, the beads look really elongated, and that's because we are actually doing multiple colors in the frame, and so that's chromatic aberration. And the size and the spread of the beads and the shape of them here helps us get the 3-dimensional information, with a little computation. So that's kind of what a bead would look like. But if we start to image a tumbling bacteria, so tumbling is code words for it's dead, but it's in water and floating around. Which I had to learn all these words. I was like, why is it tumbling? So tumbling, it means it's free to move around, but it's actually not. This cell is not actually alive. But for the single beads, you get this the defocus information that helps us. You can kind of visualize how you'd analyze the beads, and so for single particle tracking, it would be easy to think of the algorithms you design. But here you get lots and lots. You can think of this as a bunch of lights all together, and so you don't have that same information. So we need to figure out how are we going to get three-dimensional information from the bacteria. So what are our methods? So roughly speaking, we tossed out this method and I couldn't really find a good slide. But what we have for a number of different beads, we have the shape of these, and we could actually, from the beads, you could brute force figure out from calibration of the camera where the beads were in three-dimensional space. And this is roughly analogous to what a lot of the particle tracking methods in super resolution do. What's cool here, so we tracked a bead moving around in water diffusing, and we don't really have ground truth because we had a bead in the microscope. But what we noticed that was kind of interesting is you kind of see this bead, it jumps, and we actually worked out based on depth. It's hanging out on one cover slip, and then it jumps to the other cover slip. And based on distance, we think it's probably doing a fairly good job. So we can do single particle tracking, but that really wasn't what we were interested in. So we want to be able to get three-dimensional information of these other structures like cells. And for those of you who've done microscopy, usually you'd get the point spread function of, which is the forward optical model of the microscope, and then you have this inversion process. The problem here is that what we really have are, in this case, now that we're up to a 5x5 tile, we have 25 PSFs. And so we have a much more complicated, so you can think of the image formation model crudely here as you have a sample volume with something in it. You go through 25 different optical systems to produce 25 images. And so you can't just stack up these 25 images into a Z-stack and do a deconvolution like you would do with a confocal microscope or other optical system. Because if you took the 25, each slice has a different optical system creating it. So you need to be a little bit more careful about how you do this. So mathematically or computationally, we have this 3D object. We have this microscope model, and it produces a 2D image. So this is a classical ill-posed problem or inversion problem. We have the image. We know something about the microscope. We need to figure out what is F. And so one of the things I'd like to point out here is that the 3D object and the shape, the number of voxels and the dimensions, isn't really specified. As we developed our algorithms, we get to specify that, and that's largely related to the resolution we're able to pull off. But in terms of size, the image is a 1K by 1K image, and we're really trying to reconstruct a dimension, let's say 200 by 200 by 200 voxels. So it's a lot of missing information when you only have 1,000 by 1,000, and you're going to try and pull off hundreds by hundreds by hundreds. Where does that information come from? And so we use regularization techniques and optimization, and we say, well, roughly speaking, we take the best guess for initially 0 for what is the function, push it through the forward model, do a comparison, loop around, and repeat, subject to some sort of priors. So I won't talk a lot today, but those of you in optimization space, we look at sparsity priors, sparsity in the gradient space, smoothness priors, and we get different but comparable results with all the different priors we choose. Based on, I showed you earlier a movie, from each slice of this space, we can pull off a 3D volume. And here is, from that movie I showed earlier, this is again, and again, if you look at what's kind of fun to look at is, initially, the bacteria must be bumping up against one of the surfaces, and then eventually at around frame 30, it moves away, and then it starts to be able to rotate and twist. And what I'm showing here is the Z position of this is Z if you can't read the axis, and this is elevation at azimuth. And we're not really able to get that other dimension very robustly out of our data. So this is three-dimensional volumetric data of a tumbling bacteria. So then the next question we ask, because this is the sort of thing you do, is how do we know that we're right? Because we're looking at this under if you saw the data, it looks really icky. How do we know that we have something correct? And I'll give you a few examples of how we did our validation step. So what we did is we actually took fixed bacteria, we took a confocal stack, and we used that as our reference volume, and we used all sorts of things that you do. You add noise, you push it through the forward model, and that allowed us to do things like look at the impact of noise. For the optimization folks in the room, we have all the different assumptions we made, the priors, our regularization parameter, and we're allowed to explore the space and figure out how well things worked. But the advantage here is that then we can do a voxel by voxel comparison of photons in and photons out. So this is the volume that you had, the confocal stack, and what you see here is in this voxel by voxel comparison, you possibly can read the scale, but it's looking at the intensity error. And if it was perfect, it'd be just a straight line here. And it's not perfect, and we tend to get this odd shape that we're still trying to analyze why we always get this shape where it drops off. But we can look at a voxel in and a voxel out and ask, did we get there? So we're actually fairly confident that assuming you pick the right priors and the right parameters, we can do a pretty good job of reconstructing the 3D volume. And again here, these are just taking the volume and slicing it through to show the different planes, partly because biologists really like to see things spread out in pieces. I like the 3D models, and this is actually the reconstruction error. So you can see that around the boundaries, we tend to have some error in how we're computing our photons. And then we looked at does the position matter? So this is a long Z, things in one direction. That was a double low bacteria. So this is one that's probably likely about to split into two cells before it was rudely fixed. And this is just one that we noticed initially that there was some anomalies if things were in a Z direction. Okay, and then, it's biology. One tumbling bacteria is not all that interesting, so we actually started looking at larger volumes. Again, in order to do an evaluation here, this particular set of cells are fixed and, what do they call it? Plasticized, so that we can take a confocal. And here, the MFM, it took half a second, which is a lot higher than frame rate, but because we have these periplasmically labeled bacteria, so this inside of the cell is the cytoplasm. These cells are, the fluorophores hang out in the periplasm, the outside part of the cell, and that, it's a lot dimmer than the one that was lit in the cytoplasm, so we actually had to acquire for a longer period of time to really get any signal. But this is 10 seconds of confocal, and so here what you're looking at is this red line here, sliced through, looking at just the voxel values. And you'll see that we do a really good job of being able to pull out the two sides of the periplasm compared to confocal. And so we think this is pretty compelling evidence. And then this is, because it's fixed, we couldn't help ourselves. We took multiple frames and averaged them, and then we get really nice data that it's really clear that this is periplasmically labeled bacteriums. And so this is multiple cells within a single frame, all well reconstructed with our approach. Okay, so that gets us the 3D volume, and now going to 4D, so things are moving in time. What I showed earlier was we took the volume, there's an imaging model, and we do some optimization, and we reconstruct. And so every frame we can do that independently. And so if we want to go to a time series, we can think of having n different frames, each of them a 1K by 1K image, and we can just crank this machinery in every frame. And the results I showed you earlier of that colored bacterium, that was performed this way. And that's pretty computationally intensive, but it works. So we also started asking, could we make other assumptions? And if you assume over a number of frames the bacteria is roughly a rigid body, we could actually reduce the parameter space and use frames nearby, assuming that the bacterium's moving, and try and reconstruct and see if we get any better results. And so there were two approaches. I'm not going to go through the math. We did a batch approach where we said, let's take n images and process on it, or if we take this frame and say it's a recursive algorithm where you look, this frame is a motion from the previous frame. So all the results I'm going to show you are from the recursive approach. But here's so what we did, again, we took the confocal microscopy sample, and then we could control the the motion of the bacterium. So we simulate a moving object so that we could see how well our algorithm worked compared to just doing the raw single frame at a time. So here's a single frame reconstruction with our roughly best in class set of parameters so far, and the multi-frame. And again, you get slightly better peak signal to noise ratio when you do the multi-frame. Anyway, so here we have, again, single frame, just using one frame at a time to reconstruct versus using multiple frames. And we think we get slightly better measurements that way. And now on real data, so here is, again, an example. I won't go through the whole movie because it's really kind of dull. It's much more fun to see it as a moving bacterium. And if we do single frame reconstructions, we get a decent result, but we get slightly better results when we look at multiple frames at a time. And so just taking a snapshot, frame 9 and frame 42, we think that the multiple frames, we believe, is giving us slightly, it's giving us better noise statistics at least. Okay, so here is hot off the presses. So this is relatively new because we had to redesign the DOE to work on the green light for the insulin cells. And this was just data we took just a couple of weeks ago at UChicago. These are insulin cells with the MFM. Again, we went back down to the 3x3 here and reconstructed the three-dimensional volume. So we're just now starting to play with what can we do with this toy that we've now made. Okay, and so it's a computer science talk, so I thought I'd give just a rough, so where do we get improvements here? And I only have 2 slides that, we looked at doing different algorithms, optimization. People in the crowd, you have a huge optimization problem here. There's various algorithms. And we've played around. We use this twist algorithm, which is, it's an iterative shrinking thresholding algorithm. And we then developed a new interior point algorithm, and we're getting slightly better performance. So, and then we're starting to do what you guys are doing here. We're starting to scale it up. But most of our focus has been on getting the technique correct. And we haven't, we've really just done some quick and dirty, let's put it on GPU. But we've so far got about a 50 times speed up by looking at improved algorithms and scaling on the GPU. Did a lot of parallel work, but this is my colleague Mark did most of this. Most of it was on things that I brushed under the carpet in the discussion, like, okay, we have these parameters in our optimization, like the regularization parameter. How do we set that? Well, we needed to run thousands of different evaluations of our procedure, and that was all set up and run by Mark using Swift, which is a workflow tool that you may or may not have learned yet. Not yet? Okay. And so we were able to do things like generate volumes of synthetic data, large volumes of synthetic data, run through various experimental parameter sweeps to evaluate how things were working. Mostly this method is what I would say shaped from defocus and sort of multi-view in that you've got these 9 planes. And in our current effort, which we've just put in a proposal, we're trying to figure out how we can do more. So what you saw were cells, and we could see periplasm, and we could see the cytoplasm. But we'd like to go now a level, we'd like to see things in a cell. And so we think the approach that will work will be to combine different techniques. And so one of the things we're looking at is looking at interferometric techniques. And I have a picture of what we set up, but a slide here sort of giving you a rough idea. It's very similar to multifocal in that you pass light through our sample here, and if I can just go through my cartoon here, it helps. And at different depths, you're going to get different phase maps, and so this is coded here with squares and circles and triangles, which all of us understand. And so at different phases, different things will be brighter. And from this, with some computation, we can very accurately reconstruct depth. And so this is work that we're just starting now. The other thing that has been used a lot in microscopy is the idea of using structured illumination. So I showed you an example before. There's commercial products you can buy, this David scanner and other things that do it for physical objects at our scale. And you can do it in a microscopic scale. What we would like to do, so we've set up and copied what other people have done so far in structure. Now we'd like to actually do structured illumination with multifocal. So we'll have 25 different tiles of structured illumination. So this is sort of work in progress and where we're headed. Okay, 3D snapshot volumetric imaging. Our current technique, it captures 3D at frame rates of the detector. So depending what we're looking at, we can go 30 frames a second, or I guess 25 frames a second for our detector. For the multiple bacteria with the periplasm, we needed a longer acquisition time because it was not very bright, and that was at 2 frames a second, but that's still reasonably fast. And we want to, we're able to capture these tumbling cells and get the resolution to see things like the periplasm. But next, we'd like to go a level lower and actually see inside the cell. So we're going to need to invoke some of these other techniques, like the interferometry and the structured illumination, to get the spatial and temporal scales we need to see inside the cell. Okay, I'm one person. This is a team of many. So there's a lot of people that have worked on this project from various aspects. Mark and Sean and I are the MCS people that have done the algorithmic development and methods for doing the 3D reconstruction. We also work with Ali Kosert and Agalos Katsalagos at Northwestern on some of the algorithmic, and Nathan, Kwan, and Andrew are 3 students that have worked with them. And at University of Chicago, Norbert Schur in the Department of Chemistry, he's the interferometry guru that we're working with that will hopefully take us to new heights. And then various postdocs, Matt, Tuan, Alan, Ite, and Shaolei, who have worked on this project. And finally, Rose Wilton is a biologist at Argonne, and she's the one that develops all the different fluorescent sensors for us and has developed the one that does the periplasmic labeling and the cytoplasm and gets us all our specimens that we take an image. So just wanted to acknowledge that it is a very large team in order to achieve this sort of new device. And it's also very interesting in terms of DOE space. I mean, they really did fund us. The objective was they wanted us to build a new imager. They didn't want us to, they weren't, I haven't given you a lot of biology. There's motivation, but I thought it was pretty fun to work on technology, computational imaging development for a biology funding agency. With that, I will address any questions. Thank you.